Hello, and welcome to today's webinar, The Impact of Substance Use on the Developing Adolescent Brain. I'm Tracy McPherson, and I will be your moderator for today's event. This webinar is produced in partnership with the Network Coordinating Office, EMERSA, and the Adolescent Expert Project at NORC at the University of Chicago. This webinar is part of the Transitional Age Youth webinar series. As you can see, there are a number of very interesting topics already in the lineup, including the epidemiology of adolescent substance use interventions for adolescents and transitional age youth, and integrating stigmatized loss and disenfranchised grief into the expert model. These and other topics in the webinar series are all going to be available to you, and you can register for any of these at the web address on your screen. You can access everything for this webinar at the website expert.webs.com/adolescent-neurobiology. You can access them now and in the future. Our team will also send you a follow-up email today, and that's going to include the PowerPoint slides, an on-demand recording, a brief survey, and instructions on how to claim your free CE certificate. And this webinar is approved for 1 CE through NADAC. You can access the PowerPoint slides through the handouts pane that's on your GoToWebinar control panel, available on your computer or a mobile device, and you can download them from the website. You can also ask questions at any point by using your chat pane through the GoToWebinar control panel, also available on both your computer or a mobile device, and answers will be posted on the website. Now it's my pleasure to introduce our presenter. Dr. Sharon Levy is a developmental behavioral pediatrician, an addiction medicine specialist, and associate professor at Harvard Medical School. She is the director of the Adolescent Substance Use and Addiction Program at Boston Children's Hospital, the past chair of the American Academy of Pediatrics Committee on Substance Use and Prevention, and the immediate past president of the Association for Multidisciplinary Education and Research in Substance Use and Addiction, or EMERSA. And over the last 20 years, she has evaluated and treated thousands of adolescents with substance use disorder, and she has written extensively on this topic. In 2016, she established the nation's first accredited pediatric addiction medicine fellowship training program at the Boston Children's Hospital. And she has conducted research to develop and test tools for identifying and addressing adolescent substance use disorders in general medical settings. And she has expertise in the integration of substance use treatment services in pediatric primary care. So with that, welcome, Dr. Levy. I'm going to turn it over to you.
So thank you, and I would like to thank EMERSA, the ATTC, and NORC for inviting me today. I am a pediatrician who specialized in developmental behavioral pediatrics as well as addiction medicine, and I am particularly interested in the impact of substance use on brain development. So I want to start out by reminding us all that the whole concept of adolescence as a distinct developmental phase is actually rather modern. Until modern times, people by the age of 12 or 13 were generally considered adults. I know that that actually seems quite strange in today's world, but it's true. We can recognize this in, for example, the Jewish bar mitzvah ceremony, when at age 13, boys and girls are recognized as adults. Well, I did a little experiment. I went to Google Image and I typed in the word adolescent, and I made a collage of a couple of the first images that came up, and you see them here. And if you sit back and look at this collage, you could really ask yourself, how is it that for so much of human history, we did not recognize that adolescents were different than, for example, middle-aged adults? It seems so obvious. Well, it turns out that the brain, at least by weight, is its full size somewhere around age 12. This is a very old slide from a publication back in the 1970s that's looking at brain weight by age. And you can see I've highlighted age 10 to 12 to show that by the time a child reaches this age, the brain is its full size and won't get any heavier. But there's still a lot of development going on. This slide is looking at brain cells under the microscope. And we can see on the left panel, I'm showing you a slide taken from a newborn. And you can see there are a few cells, few connections, pretty much what we would expect, a lot of room for growth. The middle panel is from a 6-year-old, and you can see that there have been a lot of cells and a lot of connections between the cells growing over those 6 years. When we look at a 14-year-old, see something that might be a little bit surprising because we're actually seeing fewer cells and fewer connections between them. Well, it turns out that the 6-year-old brain is perfectly configured for learning. And we know that young children can learn things very, very quickly. They're built that way, right? And we know that you want to teach a child a language, start them speaking very early. They'll learn it very quickly. But it turns out that there's a difference between a brain that's ready to learn and a brain that's proficient. So what happens between 6 and 14 is that the brain prunes or trims away the connections it no longer needs. And the connections that are left actually grow bigger, stronger, and they get wrapped in insulating tissue called myelin that makes nerve conduction even faster. So while a 6-year-old brain is perfectly ready to learn, a 14-year-old brain is actually going to be more proficient at performing tasks. Now, this pattern of development happens all over the brain in a pattern that we see in every individual, and that, generally speaking, is back to front. This is a slide of brain scans that shows mature tissue in blue and immature tissue in greens and reds. And you can see that there's this pattern of development that starts in the back and comes over to the front. And you can also see that these slides end around age 20, and there's still a fair amount of green tissue of developing cells in these scans. And we think that this process ends somewhere around age 25. And as a pediatrician, I was interested in the correlation between the areas of the brain that are actively developing and the behaviors that we typically see in children of that age. So for example, in the first couple of years of life, the part of the brain in the back that I'm showing here in blue is called the cerebellum. It's responsible for coordinating big muscle movements, and that's developing in the first couple of years of life, just as children are learning to walk. And anybody who's been around a baby of this age can really attest that they're very interested in standing up and practicing their walking. We have this nice feedback loop because the brain matures to a point where it's ready to start walking. The child stands up, gets excited by walking, wants to practice walking, wants to kind of be up and walking all the time, so much so that we call children this age toddlers. In the preschool years, one of the areas that's developing very rapidly is called the amygdala. And this is an area that's associated with emotional regulation. And that's developing just as we see children go through the phase that some people will call the terrible twos. This is the age range when children have tantrums that seem to come out of nowhere. And just as they come out of nowhere, they go out of nowhere. The adults in the room often don't know what happened, what started the crying, or what stopped it. But this is actually very typical of children of this age. And we also know that these tantrums should be disappearing by the time a child enters kindergarten. In fact, as a developmental pediatrician, we're taught that children who have these persistent tantrums into the school age years, something is really going wrong with brain development, and we need to take a look at that. In the school age years, part of the brain called the nucleus accumbens is developing very rapidly. And this is associated with an individual developing directed motivation. And that's what's happening during the school age years, where children develop their own interests. They start developing a drive to learn and to develop interest in various areas. And then finally, the last part of the brain to develop is called the prefrontal cortex. That's the seat of what we call executive functions. And those are the parts of the brain that are responsible for the skills that help us make good decisions. Things like impulse control, error correction, self-monitoring, they all have seats in that prefrontal cortex. And that's not to say that adolescents and young adults can't make good decisions. It's just to say that good decision making is developing in those years, and so they're not reliably making good decisions. Here I'm showing a photo of a young woman who's driving a car and texting at the same time. So roughly speaking, we can define adolescence as the period in life after the part of the brain called the nucleus accumbens, or that pleasure and reward center that acts as the gas pedal of behavior, is fully mature, but while the prefrontal cortex, the executive functions that we can think of as the brakes of behavior, is still developing. And this normal imbalance is responsible for what we typically associate with adolescence, which is extreme interest in extreme behaviors and risk-taking. So this is a slide that examines what happens in the brain when individuals get rewards. So in this study, investigators brought in participants of different ages. They had them do a simple task and then receive a reward. And they looked at what was happening in their brain as they received the reward. And you can see that the participants of different ages are shown here in different colored lines. So the youngest group, the 7 to 11-year-olds, are shown here in blue. And for children, whether they got a small reward or a large reward basically didn't matter. Their brain fired about the same. And so this is why your pediatrician will tell you, if you're a parent of a young child, that if you're going to reward your child, give them a sticker, because it really doesn't make much difference in terms of brain activity, whether a child gets a sticker or a $20 bill, pretty much going to have the same level of activity. This explains why pediatricians' offices are always filled with lots of stickers. The adults are shown here in green line, and what we see is a small reward. We get a small positive blip in brain activity. A large reward, we get a larger positive blip in brain activity. Now the black line is adolescence, and what we see is really interesting. So if when they receive a small reward, their brain actually deactivates below baseline. But look what happens with a large reward. They get a lot more firing than either children or adults. So again, this drives behavior. Adolescents will be naturally looking for stimulation. They'll be naturally looking for things that will cause a lot of firing in the part of their brain, in that their reward center part of their brain. And that explains why adolescents and young adults are the part of the population that are going to be most interested in substance use, because one thing that all psychoactive substances have in common is that their final common pathway is signaling in that pleasure and reward center. So essentially what they're doing is they're hijacking the natural reward system. Instead of going through the normal physiologic pathways that result in reward, for example, when you get an A on a test or score a winning basket, your brain would normally go through a complex set of reactions resulting in firing in that pleasure and reward center. When you use drugs, the drugs will actually trigger the pleasure and reward center directly, causing a lot of firing. So if adolescents and young adults are the part of the population that are going to be neurologically driven to look for this, they are going to be the ones who would be most likely to use substances. And so if as a society we make substances available to them, we shouldn't be surprised if that's the part of the population that's most interested in using them. And so in summary, adolescents are developmentally primed to use drugs. And the data actually bears that out. This is a slide from SAMHSA that shows us the rates of initiation of substance use. And you can see that most of the action is between 16 and 20 years of age, and it falls off dramatically after that. And after age 26, very few individuals will initiate drug use. It's just not as interesting to adults. There's another point that I want to make, and that is that adolescents are also developmentally vulnerable to develop substance use disorders. So we know that addiction or loss of control over drug use is the result of changes in the pleasure and reward center, and that those changes are caused by direct stimulation by drugs. But the prefrontal cortex, those higher parts of the brain, actually can protect the pleasure and reward centers. And in older individuals, it does. But in adolescents and young adults, because that part of the brain isn't fully developed, it doesn't offer a lot of protection. You think of it like this building, the roof isn't up yet, and so it's not going to offer a lot of protection to the structures down below, not yet. So now I want to switch gears a little bit and talk about individual substances. And I'd like to start with THC, which is, of course, the active ingredient in marijuana. Here I'm showing you a picture of two different molecules. On the left, I'm showing you anandamide, which is a molecule made by the human body, which binds to brain cells or neurons and causes signaling. And THC on the right in purple, which is a molecule made by the marijuana plant. That's similar enough to anandamide that if a person ingests THC, gets it into their body, THC can actually bind to the receptors where anandamide would normally bind. And when those receptors are bound, whether by THC or by anandamide, what they do is they stop the cell from communicating with its neighbors. In other words, they quiet down cell activity. And this is a slide of the brain showing us in purple areas of the brain that are most densely packed with the cannabinoid receptors. So this part in the center that's bright purple, that is the nucleus accumbens. That is the pleasure and reward center. And we can see that that's got a lot of cannabinoid receptors, and that's what makes marijuana use feel good to people. They're getting firing in that pleasure and reward center. But there are other parts of the brain that are rich in cannabinoid receptors as well. For example, the cerebellum, the back of the brain that's responsible for big muscle movement, is also pretty densely packed with cannabinoid receptors. So we know that THC or marijuana use is going to interfere with our coordination. This part of the brain in the middle is called the hippocampus, and it is also got a lot of cannabinoid receptors. It's a part of the brain that's responsible for long-term memory formation, and so it's very important for learning. And so we can see that THC is going to interfere with learning. And then, of course, the prefrontal cortex, which is the top of the brain here, is also densely packed with these cannabinoid receptors. So we can guess that THC or marijuana use is going to interfere with good decision making. Now, there is a difference between THC and anandamide, and that is that THC is about twice as potent as anandamide. In other words, if both anandamide and THC are both near a neuron, the THC is more likely to get into that receptor, and it'll also sit there longer, about twice as long on average. And this is important because we talked earlier about how the brain develops by pruning, by removing the cells that are quiet. So if we have THC sitting in receptors, because somebody's been exposed to marijuana or some sort of THC compound, it's going to alter the shaping of the brain and the pruning of the brain. We want pruning to be done with surgical precision, and so I'm showing a scalpel on the left. We don't want it to be done by a blunt instrument, and so that's why it's so important to avoid marijuana use during adolescence and young adulthood while the brain is still developing. Now this is another slide with a different view of the hippocampus shown here in blue, and it, as demonstrated a few slides back, it is rich in cannabinoid receptors, and it's been shown that people who use marijuana heavily during their adolescence actually on average have smaller hippocampuses than their peers. And it's also been shown in this study from about 8 years ago that was done in New Zealand that persistent cannabis use actually is associated with declines in neuropsychological testing. So in this study, the researchers recruited more than 1,000 children at age 13 before marijuana use had started, and they caught up with them every 5 to 6 years, and they administered them a bunch of questionnaires and also an IQ test, and they just watched what happened over time. And what they found was that when they looked at the group of people who reported no marijuana use at any of the time points, they saw that the IQ was stable, as we would expect. So with an average of around 100 without change over that 25-year period. But when we look at the people who had used marijuana most heavily, who met criteria for what we used to call marijuana dependence on 3 or more occasions, they actually had an IQ change of about 6 points over the course of that 25-year period. And this was statistically significant change compared to never users. So can say by looking at this that marijuana use during the adolescent years is associated with a decline in IQ. Now, I want to point out that a 6-point fall in IQ is not something that you'd be able to pick up with casual conversation. This was only picked up with neuropsychological testing. But it is a signal to us that, gee, maybe something is really going wrong when people use marijuana during those adolescent years. Okay, I want to switch gears again, and this time talk about nicotine, which is a molecule that's made by the tobacco plant. Here I'm showing nicotine on the right. On the left, I'm showing a drawing of acetylcholine, which is a chemical made by the human body. And nicotine, just like THC can mimic anandamide, nicotine can mimic acetylcholine and bind to acetylcholine receptors in the central nervous system. Now, nicotine actually is a very avid binder to that acetylcholine receptor, and very quickly it actually starts making changes in neurons. So here you're seeing a schematic. On the upper left, we see a non-smoker. This is what the cell is supposed to look like. You can see in the middle panel, we have a new smoker, and you can see that the black dots are nicotine molecules, and they very rapidly and avidly bind to those acetylcholine receptors. They're actually out competing the body's own acetylcholine. And what they do over time is they cause changes in the cell. So the cell will have fewer receptors in order to protect itself and maintain homeostasis. Will actually have fewer receptors, and this, of course, gives rise to the tolerance that we see with exposure to nicotine, and also the withdrawal symptoms when somebody stops using, because these cells are no longer in their normal state, and they need actually nicotine around to stimulate them in order for them to function properly. So this is a slide that just shows us how quickly nicotine binds in the brain. On the top, you're seeing brain scans of somebody who is not smoking. On the bottom, you're seeing brain scans of somebody who is smoking. And this is the first slide is after just one puff, or about 1/10 of a cigarette. By the time somebody gets to 3 puffs, their brain is almost completely saturated. By the time somebody's had 3 complete cigarettes, you can see everything is completely saturated. So that actually happens pretty quickly. And this is a schematic just showing us the projections in the brain. So what are the parts of the brain that nicotine impacts? And of course, just like all substances that have psychoactive properties, there is going to be firing in the nucleus accumbens related to nicotine use. And there are also projections onto that prefrontal cortex, right? Onto the parts of the brain that help with executive functioning or in control of executive functioning. And there are also important differences in how adolescents and adults experience nicotine. And I've shown you a diagram that shows some of the places in where the experience of an adolescent is exactly the opposite to the experience of adults with nicotine stimulation. And this certainly has functional correlates. This is looking at problems that are associated with smoking. So we're looking at in black, these are 7th graders who are considered smokers. The light gray is 7th graders who've tried smoking but aren't regular smokers, and then in the other the white box, we're seeing non-smokers. And you can see no matter what problem we look at, we see a scale where those who use nicotine have more of the problem than those who haven't. And a lot of this work was done looking at people who use traditional cigarettes, but these days, particularly when it comes to adolescents, we see much more vaping than we do traditional nicotine use. And that's really important because the way that nicotine is delivered by the vape, because the vapor that comes out of the vape that carries the nicotine can be inhaled far deeper into the lungs. And because the concentrations of nicotine can be much higher in vape liquids than they are in traditional cigarettes, people who use pods can actually get much, much higher doses of nicotine than people who smoke traditional cigarettes normally would. So this is from a publication in Pediatrics that was looking at urinary cotinine concentrations. Cotinine is a metabolite of nicotine. And what we see is when people used tobacco, they had a mean of about 330 nanograms per milliliter of cotinine in their urine. E-cigarette users, so those are the initial e-cigarettes, actually had somewhat lower concentrations, but when we look at pod users, so these are the people using Juuls and other pod devices, their cotinine concentrations were nearly double. And so not only can the device deliver a much higher dose of nicotine, but it's also the case that the way in which adolescents typically use these devices also encourages greater absorption and higher peaks of nicotine. So I've put here a dictionary. These are all of the words that I've heard my adolescent patients use to describe how they're using their vapes, and often I have to just write it down and go look at YouTube later to figure out what they're doing. But these are all tricks that kids will describe using these vape devices, and all of them encourage deeper inhalation, greater inhalation of vapor, and holding the vapor longer, all of which are related to higher doses of nicotine being delivered. And in fact, it's very common for kids who use these devices to describe themselves as being nic sick, which means that they're feeling nauseated, dizzy, or having a headache, which are all signs of nicotine poisoning. In fact, these symptoms are very common amongst kids who use these devices, and that actually as a clinical finding is really very striking. So it's not uncommon for kids to get so much nicotine that they're actually having some toxicity from it. And this is just to underscore the differences between adolescents and adults. So this is a quote from a 14-year-old boy who I saw in the clinic. He was using a vape pen. And when I asked him what it felt like, he said, when I first used, I would get a buzz and my entire body would feel tingly. My eyes would water up. I would feel as if I had been spinning around and had just stopped. And to me, I thought he was initially describing how terrible it felt, but then he continued on and he said, it felt great. And I think that's all you really need to know about the differences in how a younger person experiences this compared to an older person. And then he went on to say, I don't feel those things anymore. And of course, he was describing the tolerance that he had developed over time. Okay, and switching gears one final time, I want to end with some comments about alcohol. And here is a picture of alcohol. It's actually a pretty simple molecule. In fact, for anybody who remembers some basic chemistry, it actually looks a little like the water molecule, and it actually has water-like properties in the body in that it can bind to a lot of places, unlike THC and nicotine, which have specific receptor targets. Alcohol actually has many binding targets, some inside of cells, some outside of cells, and it can actually have a real mix of actions, a little bit more than typically other drugs do. Here I'm showing you on the right another picture of the alcohol molecule. On the left, I'm showing you a molecule called GABA that is, of course, made by the human body. And most of alcohol's effects actually take place at the GABA receptor in the brain. Here I'm showing you a schematic of what that receptor looks like. You can see in green, the place where GABA would bind is a little different from the place where alcohol binds. Just as an aside, benzodiazepines also bind to the GABA receptor, as do barbiturates. So it's a lot of drugs actually have their effects or their primary effects through the GABA receptor. And this is a schematic showing us the projections of alcohol on the brain. And as with THC and as with nicotine, we know that there are going to be projections to the reward center. There's going to be firing, dopamine release in that pleasure and reward center, and that's what makes alcohol use feel good. There are also projections to the prefrontal cortex, which means alcohol use is going to impact our planning and decision making and judgment. And there are also projections to the amygdala, or the emotional control area of the brain, showing us that, or reminding us really, that alcohol is going to have an impact on our emotional regulation. Now, as with nicotine and marijuana, the effects of alcohol are really quite age dependent, and the projections to the nucleus accumbens are much more relevant in younger people. And so much more than adults, younger people get a dose response to drinking alcohol. So the more they drink, the more stimulation they're going to get at the nucleus accumbens. And that actually has a behavioral correlate, because 90% of all alcohol consumed by underage drinkers occurs in the context of a binge. So this statistic alone tells you everything you need to know about adolescent drinking. Adolescents drink in big bursts. They binge drink. That is the most common pattern for adolescent alcohol consumption. This is really quite different from adult drinking that can vary, although for many adults drinking is more moderate, with only 1 or 2 drinks consumed per occasion, although of course there is a big range, and plenty of people, and especially those who started drinking younger, will continue to binge drink into adulthood. Now, alcohol is more complex in some ways than other substances, because it does have more than one target. And I'm showing here in yellow that alcohol can also mimic glycine, and that'll have its greatest impacts in the brain stem and spinal cord. And actually, the effects here are also quite age dependent. So, in fact, what we see is a lot more inhibition of motor activity among adults with alcohol use compared to adolescents. And this is actually also true in rat models. And so here is an experiment that really demonstrates it, that was done with rats. So it turns out that rats can swim, but they don't like water. So if you put them in a pool of water, they will very quickly try and swim onto a platform and get themselves out. Now, if you take an adult rat and before you put him in the water, you get him a little drunk, what's going to happen is when he swims, it's going to take him a lot longer to find the platform because his movements are so much slower. And really, this is his motor coordination is being impacted, it's being depressed by the alcohol. And because he's swimming so much slower, it actually takes him much longer to get to the platform. Now, if you compare that to an adolescent rat who you get a little intoxicated before putting him in the water, and what we see there is that the adolescent rats actually will swim at the same speed, whether they've had alcohol in their system or not. That's because they have much less motor depression with alcohol compared to adults. It's just impacting them very differently. But it still takes adolescent rats much longer to get to the platform because they simply lose their way. So they actually can become a little activated when they have alcohol in their systems. And in humans, this has correlates too. So adults, when they've had too much to drink, are much more likely to become sleepy and inactive, whereas younger people are much more likely to be active, to engage in sexual activity, to wander outside of a house and get lost, or to drive a car. And actually, this is a graph showing us the relative risk of a fatal car accident by blood alcohol concentration, and the different lines show people of different ages. And really what it's showing you is that at every blood alcohol concentration, the younger you are, the much greater your risk. And this is, again, because of that combination of there's no impact on motor function, but there is impact on coordination and decision making and all of those other things. And so I will just take a moment then to summarize the major points that I hope the audience will take away. The first point is that adolescent development primes teens for substance use. They are an age group that is looking for stimulation in that pleasure and reward center. They are naturally evolved to look for that kind of stimulation. And so substance use is a very efficient way for them to achieve that. If we make substances available, generally speaking, adolescents and young adults are the part of the population who are going to be most interested in using them. The second point is that all psychoactive substance use triggers dopamine release, either immediately or with nearly immediately with a short chain of events. And without the protection of fully developed frontal lobes, adolescents are at greater risk for neurological changes that are associated with addiction, which really talks about the loss of control over substance use and the use of substances even beyond the point in which they're fun or stimulating to the person using them. And finally, just to point out that each substance has unique impacts on other areas of the brain, and so that leads to the unique clinical picture resulting from substance use disorders and from long-term use. But all of the substances have particular impacts on brain development, and this is the reason that substance use is really quite detrimental to the developing adolescent brain. And with that, I thank you for your attention.
Thank you, Dr. Levy, and thank you for sharing your expertise with us and in making what is a complex topic of neurobiology very understandable and very helpful to those of us who are really trying to understand that connection. So thank you very much for that, and I really look forward to being able to share what you've presented today with the folks that are going to attend our webinar series. And I think this webinar really complements the other topics in the series. So we'll be building on this and talking about the epidemiology of adolescent substance use, and then talking about the interventions that are appropriate for adolescents. And so this really gets us started, and what a wonderful presentation. In our last few minutes together, I'd just like to remind everyone who's with us today that our team will send you a follow-up email later today, and this will include a link to the PowerPoints that Dr. Levy shared with you. We're also going to put the on-demand recording up there as well, and you're going to receive a link to a very brief survey, and we would really appreciate it if you could complete this and give us, share with us your feedback. And in addition to that, the email is going to include instructions on how to claim your free CE certificate. So all of this will be not only in the email, it will also be posted to the website at expert.webs.com/adolescent-neurobiology. So if you want to access anything, you can access from there. If you have any questions about the resources, you can certainly email us as well. Also, I'd like to share with you some related products and resources that are available from the ATTC network. As you all probably know, there's a robust set of resources that are available from the ATTC network. Here is just a sample of those that are on demand and ready for you to access 24/7. It includes the presentations and the handouts. And so we hope that you'll take advantage of these and share them with your colleagues. As you can see, there's quite a diverse set of resources on class standards in behavioral health, understanding Latino youth recovery, more on the adolescent brain maturation and health. And as you can see, there's additional ones that are sort of topic specific to a substance, so marijuana use specifically, and those affects a little bit more detail on the developing adolescent, and vaping, which we all know is a really important and growing topic and is getting a lot more attention. And so there are multiple resources on that topic. In addition, there is a recording available to you on understanding suicide and adolescence and the changing brain. So I hope you'll take advantage of all of these. You can access all of them from this web address on your screen. In addition, I hope that you will not only follow us and take advantage of these resources and the webinars that we shared with you that are coming in the future, but also we'd like to keep in touch and stay connected with you. There are a number of ways that you can do that. The ATTC Network office has a monthly publication called The Messenger, and you can very quickly subscribe and get the latest news and resources that the ATTC network has developed. You can also connect with the ATTCs on Twitter and Facebook at ATTC Network. And so, again, visit the website and you can follow and sign up and see the latest newsletters and information from the ATTCs. Also brought to you another whole host of resources from the PTTC. And so this network has, as you can see, a number of on-demand learning and webinars that even extend some of the topics that I just shared with you available from the ATTC. So there's a series that talks about underage alcohol use, also expanding into opioid addiction, and then selecting and implementing those evidence-based practices to address substance use among young adults. And there are resource guides that are available to you, and they're easily downloadable. There is also a two-part webinar series, again, extending the vaping topic, and around this one particularly is around the extent and risk factors for youth vaping. And so you can see there's part 1 and part 2, and part 2 really talks about the policy recommendations and the promising practices to address vaping. In addition, you can see here that there are a number of diverse topics around the benefits of engaging youth in communities. And also a 6-part webinar series, which is from the Mountain Plains PTTC, on informing prevention. So in addition to all of these resources, you can not only listen and learn, but you can download and share, and we hope you'll take advantage of those, all available from one resource. And we'd like to stay connected to you through the PTTC as well. So there is a monthly publication called the PTTC Post, and you can subscribe directly to that through the web address on your screen. And you can stay connected also through Twitter and Facebook using the @PTTC Network and the @PreventionTTC Network. So again, please take advantage of all these resources, and we want to stay connected with you. So with that, I just want to thank everybody so much for attending, and thank you, Dr. Levy, for sharing your expertise. And with that, I'm just going to conclude today's webinar. Thanks so much, and we look forward to having you on a future webinar.